Hello viewers, welcome to Biodrunks. I'll be providing notes, PDF link in the description.
In the previous video, we did more details about cosmid, phagmid, and phasmid. Okay, so these three were the most important topics which we did in the previous class. And in today's video, we are going to do something about artificial chromosome vectors. So let's learn what is this artificial chromosome vectors. So when I say artificial chromosome vectors, so these are autonomously replicating DNA sequences, and they contain centromere, then they have two telomeres, and they have origin of replication. So these are the most important things which are present in the artificial chromosome vectors, and because of these sequences, they are being used for cloning of DNA inserts of large size. So now you know what is this artificial chromosome vectors. Let's learn the types. So this artificial chromosome vector is of different types. Bacterial artificial chromosome, we also call it as PAC. P1 derived artificial chromosome, we call it as PAC. Yeast artificial chromosome, we call it as YAC. And mammalian artificial chromosome, that is MAC. And human artificial chromosome, that is. So mammalian and human artificial chromosomes, we are going to do it together. Okay. And bacterial P1 derived yeast, we will do it separately. Great. So let's learn the first one, that is bacterial artificial chromosome. So when I talked about a bacterial artificial chromosome, okay, so this has its own origin of replication from E. coli. Okay, so this origin of replication from E. coli has an F factor. So this F factor allows strict copy number. Please remember this F factor allows strict copy number. Okay, and it maintains 1 to 2 copies of vector per cell. 1 to 2 copies. Okay, now let's learn in detail about each part here. So first is rep E. So what is this rep E? This encodes for rep protein. Okay, now this rep protein, what does it do? It binds to the ORI-S to initiate replication. Okay, so you can remember rep protein means it is initiating replication. Then you have par A, par B, par C. So these are involved in partitioning of F plasmid. Okay, so what does they do? They partition. They are involved in partition of F plasmid. Now, when does this partition occur? This partition occurs during cell division because the cell is going to divide into one more cell, two cells. Okay, that is where it is being used. Now, what is this cos n? So you know about cos site when we did cosmid. Okay, so I'm just giving a cosmid, very basic example. Cos site is present in the lambda phase. Okay, so in this cos site, it's highly important because it gives rise to sticky ends. So cos n is also present here. Now, you have resistant genes, okay, which have been used as markers. That is, here you have ampicillin resistant gene. Now, LOX P. Okay, what is this LOX P? So LOX P is a region for where recombination occurs. So it's a region where the recombination is occurring. Okay, and MCS, you already know about it, multiple cloning sites. So this was all about bacterial artificial chromosomes. So easy. Rep is for replication. Par is for partition. Okay, that occurs during cell division. Ampicillin R, that is resistant gene. LOX P for recombination. Cos N is the cos site, and MC is multiple cloning sites. Great. Very easy. We completed first topic, that is bacterial artificial chromosome. Let's go to P1 derived artificial chromosome. Okay. So what is this P1 derived artificial chromosome? So this P1 derived artificial chromosome is a DNA construct that are derived from the DNA of P1 bacteriophage. Okay. So again, it has the same site. As you can see, this is the pack site for the binding. LOX P is for recombination. ORI, origin of recombination, kanamycin resistant gene. Okay, and a replicon. What is replicon? Replicating units is called as a replicon. Okay, so this is a derived artificial chromosome. Okay, and this is used to clone DNA fragments of nearly 100 to 300 kilo base pairs in size. Okay, and this does in E. coli cells. So we are done with the second one, that is P1 artificial chromosome. Let's learn about yeast artificial chromosome. So before knowing about yeast artificial chromosome, keep in like remember yeast chromosomes or yeast vectors are of two types. One is yeast artificial chromosome and another is plasmid vectors of yeast. Okay, so in detail, I'll be dealing with yeast vectors in the next video. Okay, so in that video, I won't be talking about yeast artificial chromosome vector because I already did it here. Okay, so this yeast artificial chromosome vector, what does it have? Always remember, each artificial chromosome vector is a linear vector. Okay, it's a linear vector and it behaves like a yeast chromosome. So what does it contain? The first thing is contain is ARS, autonomously replicating sequences. Centromere, CEN, ORI, origin of replication. It has two different markers. If you can see here, these are selectable markers, ampicillin resistant gene, and these are telomeres. Okay, so this is what it contains. Okay, now the important sequences from E. coli are used here for selection, propagation, and E. coli. So these yeast artificial chromosome vector, it is used to clone very large DNA segments. And the main motive of this is mapping of complex eukaryotic chromosome. Okay, it's used for mapping of complex eukaryotic chromosome. Great. So I think this was the most easiest one, but this is the most important artificial chromosome vector and it is highly used. Okay. So this was all about, yeah. Now we are left with mammalian artificial chromosome and human artificial chromosome, which we are going to do it together. Now, what is this mammalian artificial chromosome? So, mammalian artificial chromosome, these and human artificial chromosome. Okay, MAC and HAP. These are vectors. They contain centromeres. Okay, because it's already an artificial chromosome, so it has centromeres, telomeres, origin of replication, and they have transcriptionally active chromosomal domain. Okay, so these have transcriptionally active chromosomal domain. Okay, now these are MAC and HAC. They are constructed in the cell culture labs. Okay, or I should say in vivo or in vitro by artificially changing the structure of natural chromosomes. So as you can see here, I have just kept a good example. I just got it. Here, this is a human cell in which you are taking a chromosome number 21. So this chromosome number 21 is transferred into a chicken DT40 where the recombination is occurring with a linear plasmid, if you can see here. Okay, so here with a linear plasmid, the recombination is occurring. Always remember the chromosome has two arms, P arm and Q arm. So first you have a transcription in the P arm and then you have a transcription in Q arm. Okay, now after this transcription, this is being transferred to a hamster. Okay, hamster cells. Now, always remember telomeres are what? They are repeated units. Okay, and they also have selectable markers with it in human artificial chromosome and MAC artificial chromosome. So these units are being cloned or inserted into a back vector. So you always, what happens is these mats and the yaks, they insert their vectors into yak or back. Okay, so these yak or back should have a human centromeric DNA. Okay, and then they are transfected into the human cells, as you can see here. Whatever recombinant I need, I have it. They transfected into the human cell, and you can analyze it in fish. So one video will be there regarding fish. What is fish? Fluorescent in situ hybridization. Great. So that's how it works. So whenever you create some DNA like this, generate a particular recombination like this, they are about 1 to 5 megabase pairs in size. Okay, and they contain yak or back multimers. So this was a brief explanation about artificial chromosome vectors. Okay, and in the next video, I'll be doing about yeast vectors, vectors for plants, and shuttle vectors.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.